All right, so firstly, obviously my name is Alex Gorman and I'm really honoured to be here, so thank you. And I would like to talk to you guys about the high levels of genetic diversity and gene flow found in the marine angiosperm Amphibolis Antarctica. So firstly, I'd just like to reiterate the importance of our seagrasses worldwide. They are considered to be one of the most valuable ecosystems in our biosphere, and they are used for things like wave attenuation, oxygenation of the water, sediment stabilisation. They provide nurseries and habitats for a large amount of marine biota. So, moving on, the reason we chose to study the species Amphibolis antarctica is because it is endemic to temperate Australian waters. It ranges from the subtropical coast in Western Australia round to New South Wales along the southern coastline. It also reproduces both sexually and via colonial growth. So its dioecious and viviparous nature is a very unique combination found in seagrasses, and therefore we could analyse the levels of genotypic richness across a wider scale. The propagules are released in May and June for this species, which allowed us to look at potential for propagule dispersal also. So my study objectives. Firstly, it was to investigate the population structure and population genetic connectivity of Amphibolis Antarctica meadows in the Perth metropolitan area. And then I furthermore had a broad-scale study of the genetic diversity found between my local site in Perth and compared it to South Australia. To answer these study objectives, I asked myself 4 questions. I had to figure out how colonial the populations were in the Perth metropolitan area to get a level of genotypic richness and to find out whether the meadows were reproducing predominantly sexually or via colonial growth. From there, we looked at the genetic connectivity present in the meadows, and that gave us the opportunity to figure out what direction they might be, the meadows are connected and whether they would be connected on a large scale or on a small scale. From there, we analysed the population structures individually. This was across all sites, and then I could look at whether on a broad scale, Western Australia and South Australia has similar population structures, and therefore we could look at the genetic connection or diversity between the two. Now, after all of that, I had to ask myself what the connectivity data told me about its potential for the seedling transportation in the meadows along the temperate coastline. So my population sites. My first population site was in Shark Bay in Western Australia. This is approximately 700 kilometres north of Perth. And then I had 7 population sites in the Perth metropolitan area. These stretched along 40 kilometres of coastline and extended as far out as 20 kilometres off the coast to Rottnest Island. From there, I also had 4 population sites in South Australia. These were my broad scale sites to compare with my local population. So I had 4 sites ranged along a coastline kilometre of 374 kilometres. And then I could then run my connectivity scales from there. So my methodology. For my local scale, I used a haphazard sampling method that was conducted over a period of 10 months. I had 7 population sites that I sampled from, and I picked 50 plants from each site. They were approximately 2 metres apart, and from this point I took them back to my lab, extracted the meristematic tissue from each of them, and then I dried them using absorbent tea bags using silica gel for a period of 2 weeks. From this point, they were then further dissected down to 3mm by 3mm samples and put in centrifuge jars, where they were then sent off to the University of Adelaide, where they underwent DNA extraction. The DNA extraction we used a cell disruption method, and from this point it exposed the DNA. We could then isolate the 15 molecular markers, which we then later removed one, but I will get to that. and then they were sent to the Georgia Genomics facility in the US. Here they underwent high-resolution capillary electrophoresis and fragmentation analysis, and then the university sent us data where we then ran a microsatellite scoring allele program, and this gave us usable information which we could then convert into statistical analysis, and then we could run genetic scoring and get an understanding of population structure and gene flow amongst the different meadows. So firstly, clonality. The measure of clonality provides an understanding of genotypic richness. What we found overall was that there was a high level of genotypic richness. This indicated that there was a high level of sexual reproduction, but instead of clonal growth. However, this is with excluding one meadow in Western Australia, Geordie Bay on Rottnest Island. We excluded this meadow because it was solely a clonal meadow. It was monospecific, so we believe this could have been an influence from human impact or something like that, because it was a meadow at the end of a jetty. The high levels of, it was then possible to compare the local populations and identify any genetic connectivity between the sample sites on a local scale, and then in both Western Australia and South Australia, and then furthermore compare them on a broad scale. Oh, sorry, I hit back. So this is what we found. So we found that, if you can see here, this is the Perth metropolitan area. The dark lines indicate a high level of genetic connectivity between my meadows. So here we have South Beach in Fremantle, and over here is Parker Point on Rottnest Island. This thick black line showed that there was a strong connection between the gene flow and the genetic connectivity of these particular meadows, which indicated that there was propagule dispersal in that direction, in either direction. We also found that there was a significant connection in nearly all of the sample sites that I found. There was particularly strong connection down in the southern area of the Perth metropolitan area, where we had local dispersal was indicated strongly. Overall, though, on our broad scale, we found that there was genetic connectivity present between the 4 sample sites in South Australia as a local site. However, there was a very weak connection between the Perth metropolitan area and South Australia. We believe that with further research and with further studies gradually going down the coastline and down the west coast and across the southern coast of Australia, we'd see a gradual decline in the genetic connectivity of the plants, and therefore that would explain why there's such a large genetic diversity between the 2 sample sites. So furthermore, we looked at the population sites within the population structure between Western Australia and South Australia. What you can see is we used the program structure 2.3.x, and this broke it down into different clusters of genetic information. And what we found is that we could see here there's a clear divide between the 12 sample sites that I had. The ones in orange are indicated the West Australian sample size, and then the ones in blue are the South Australian sites that I had. As we ran further testing and more clusters were formed, the West Australian samples were held strong, showing that there was a strong population structure for the 8 sample sites that we had. However, we found that in South Australia they started to break away. So the green structure is Goose Island, which was the one closest to Western Australia, and the one in purple was Robe Beach, which is the one furthest away from Western Australia. And the 2 in the middle are Merino Rocks and Henley Beach. Now, these 2 are actually quite close together as sample sites, so you could infer that potentially that because they are so close together, the population structure is similar in that respect, because they have the same gene flow between the meadows in that respect. So it would be very interesting to further look at why they broke away that way, but we didn't have the time, unfortunately. So finally, I'd just like to say why we looked at all of this. So seagrasses are a keystone species worldwide. Without them, we wouldn't have the marine biota that we have today. And by looking at the genetics and the connectivity of not just this specific species, but other different species, we can use it for accurate conservation strategies, restoration, and we could look at improving the resilience of the plants. So in respect to things like climate change, we had a strong population in Shark Bay, which is a subtropical climate, and the water temperature is quite different to what you would find in Perth. So with climate change, the water temperatures are actually going to increase in the Perth metropolitan area. So if we can harness the genetic information or the genetic DNA from the seagrass populations found in Shark Bay, we can look at restoration and transplantation of those sites to improve the survival rates of the species as a whole, and we can look at the evolution of the species as the climate changes and environmental factors change. Furthermore, we can look at it for the invasive species. By understanding the genetics of invasive species, we have a way to reduce it and let the and then promote the growth of native species again. There's been studies done on Posidonia, looking at the connectivity in Posidonia, and it was important to find to gather different data on different seagrass species. So by choosing to look at analyse Amphibolis Antarctica, we could broaden the overall understanding of genetic connectivity between and gene flow between the seagrass species worldwide. Thank you, guys.
Gary.
Hi.
I was really intrigued. Thank you. It was a great talk.
Thanks.
I was really intrigued with the very, very strong connectivity you got between your rock farm site and all the coast, not just directly.
Yeah.
Do you think there's any way of pulling apart whether that's actually like a 7,500 year signature of rockets being wet when the rest of that coastline was dry, and we've had sea level rise subsequently, or from the actual connectivity in terms of ecological connectivity now?
I don't really know, to be honest, but it would be something very interesting to look at. We were trying to look at the potential direction of the flow and trace the ancestry in that way, but it's obviously very hard for us to trace direction without monitoring the propagules. So listening to Alana talk before was very interesting for me, because it would be something that I would be interested in trying for temperate waters rather than what they did with the tropical coast. So it would be interesting for me to, further research that. Sorry, I can't answer it, though.
We've got time for one quick question.
Thank you. It's very interesting about this, because I'm doing similar, not exactly the same, but similar. But so I just want to know, is there any genetic evolution with your specimen analysis, research analysis?
So we didn't look at we traced the genetic connectivity rather than the individual DNA of each species, because I had so many samples to process and it was only my honours research and had a very short amount of time to do it. However, Michelle, who is one of my supervisors, is there, and my other one, Jennifer, they are continuing the research, so they might look into something like that at a later date.
Okay, we'll have to
Yep.
stop it there. Thank you.
Thank you very
Alex.
much.